Vision. The eye. 2. Receptor and neural function of the retina. Guyton and Hall Textbook of Medical Physiology. Chapter 50, pages 626 to 638. The retina is the light-sensitive portion of the eye that contains 1. The cones, which are responsible for color vision, and 2. The rods, which are mainly responsible for black and white vision and vision in the dark. When either rods or cones are excited, signals are transmitted first through successive layers of neurons in the retina itself and, finally, into optic nerve fibers and the cerebral cortex. The purpose of this chapter is to explain the mechanisms by which the rods and cones detect light and color and convert the visual image into optic nerve signals. Anatomy and function of the structural elements of the retina. Anatomy and function of the structural elements of the retina. Layers of the retina. Figure shows the functional components of the retina, which are arranged in layers from the outside to the inside as follows. 1. Pigmented layer. 2. Layer of rods and cones projecting to the pigment. 3. Outer nuclear layer containing the cell bodies of the rods and cones. 4. Outer plexiform layer. 5. Inner nuclear layer. 6. Inner plexiform layer. 7. Ganglionic layer. 8. Layer of optic nerve fibers. And 9. Inner limiting membrane. After light passes through the lens system of the eye and then through the vitreous humor, it enters the retina from the inside. See figure. That is, it passes first through the ganglion cells and then through the plexiform and nuclear layers before it finally reaches the layer of rods and cones located all the way on the outer edge of the retina. This distance is a thickness of several hundred micrometers. Visual acuity is decreased by this passage through such non-homogeneous tissue. However, in the central foveal region of the retina, as discussed subsequently, the inside layers are pulled aside to decrease this loss of acuity. Anatomy and function of the structural elements of the retina. Layers of the retina. Figure shows the functional components of the retina, which are arranged in layers from the outside to the inside as follows. 1. Pigmented layer. 2. Layer of rods and cones projecting to the pigment. 3. Outer nuclear layer containing the cell bodies of the rods and cones. 4. Outer plexiform layer. 5. Inner nuclear layer. 6. Inner plexiform layer. 7. Ganglionic layer. 8. Layer of optic nerve fibers. And 9. Inner limiting membrane. After light passes through the lens system of the eye and then through the vitreous humor, it enters the retina from the inside. See figure. That is, it passes first through the ganglion cells and then through the plexiform and nuclear layers before it finally reaches the layer of rods and cones located all the way on the outer edge of the retina. This distance is a thickness of several hundred micrometers. Visual acuity is decreased by this passage through such non-homogeneous tissue. However, in the central foveal region of the retina, as discussed subsequently, the inside layers are pulled aside to decrease this loss of acuity. Foveal region of the retina and its importance in acute vision. The fovea is a minute area in the center of the retina, shown in figure, occupying a total area a little more than 1 square millimeter. It is especially capable of acute and detailed vision. The central fovea, only 0.3 mm in diameter, is composed almost entirely of cones. These cones have a special structure that aids their detection of detail in the visual image. That is, the foveal cones have especially long and slender bodies, in contradistinction to the much fatter cones located more peripherally in the retina. Also, in the foveal region, the blood vessels, ganglion cells, inner nuclear layer of cells and plexiform layers are all displaced to one side rather than resting directly on top of the cones. This allows light to pass unimpeded to the cones. Figure. Photomicrograph of the macula and of the fovea in its center. Note that the inner layers of the retina are pulled to the side to decrease interference with light transmission. Foveal region of the retina and its importance in acute vision. The fovea is a minute area in the center of the retina, shown in figure, occupying a total area a little more than 1 square millimeter. It is especially capable of acute and detailed vision. The central fovea, only 0.3 mm in diameter, is composed almost entirely of cones. These cones have a special structure that aids their detection of detail in the visual image. That is, the foveal cones have especially long and slender bodies, in contradistinction to the much fatter cones located more peripherally in the retina. Also, in the foveal region, the blood vessels, ganglion cells, inner nuclear layer of cells and plexiform layers are all displaced to one side rather than resting directly on top of the cones. This allows light to pass unimpeded to the cones. Photoreceptor cell. A photoreceptor cell is a specialized type of neuroepithelial cell found in the retina that is capable of visual phototransduction. The great biological importance of photoreceptors is that they convert light, visible electromagnetic radiation, into signals that can stimulate biological processes. To be more specific, photoreceptor proteins in the cell absorb photons, triggering a change in the cell's membrane potential. There are currently 3 known types of photoreceptor cells in mammalian eyes: rods, cones, and intrinsically photosensitive retinal ganglion cells. The 2 classic photoreceptor cells are rods and cones, each contributing information used by the visual system to form an image of the environment, sight. Rods primarily mediate scotopic vision, dim conditions, whereas cones primarily mediate to photopic vision, bright conditions, but the processes in each that supports phototransduction is similar. A third class of mammalian photoreceptor cell was discovered during the 1990s, the intrinsically photosensitive retinal ganglion cells. These cells are thought not to contribute to sight directly, but have a role in the entrainment of the circadian rhythm and pupillary reflex. Rods and cones. Figure is a diagrammatic representation of the essential components of a photoreceptor, either a rod or a cone. To the right in figure are labeled the major functional segments of either a rod or a cone: 1. The outer segment, 2. The inner segment, 3. The nucleus, and 4. The synaptic body. The light-sensitive photochemical is found in the outer segment. In the case of the rods, this is rhodopsin. In the cones, it is one of 3 color photochemicals, usually called simply color pigments, that function almost exactly the same as rhodopsin except for differences in spectral sensitivity. Figure. Schematic drawing of the functional parts of the rods and cones. This scanning electron micrograph shows rods and cones in the retina. This color scanning electron micrograph shows rods, green, and cones, blue, in the retina. Fluorescence micrographs of retinal rod and cone. Rods and cones. As shown in figure, the outer segment of the cone is conical in shape. In general, the rods are narrower and longer than the cones, but this is not always the case. In the peripheral portions of the retina, the rods are 2 to 5 micrometers in diameter, whereas the cones are 5 to 8 micrometers in diameter. In the central part of the retina, in the fovea, there are rods, and the cones are slender and have a diameter of only 1.5 micrometers. Figure. Membranous structures of the outer segments of a rod, left, and a cone, right. Note in the outer segments of the rods and cones and the large numbers of discs. Each of the discs is actually an enfolded shelf of cell membrane. There are as many as 1,000 discs in each rod or cone. Both rhodopsin and the color pigments are conjugated proteins. They are incorporated into the membranes of the discs in the form of transmembrane proteins. The concentrations of these photosensitive pigments in the discs are so great that the pigments themselves constitute about 40% of the entire mass of the outer segment. The inner segment of the rod or cone contains the usual cytoplasm with cytoplasmic organelles. Particularly important are the mitochondria. As explained later, these mitochondria play the important role of providing energy for function of the photoreceptors. The synaptic body is the portion of the rod or cone that connects with subsequent neuronal cells, the horizontal and bipolar cells, that represent the next stages in the vision chain. Pigment layer of the retina. The black pigment melanin in the pigment layer prevents light reflection throughout the globe of the eyeball. This is extremely important for clear vision. This pigment performs the same function in the eye as the black coloring inside the bellows of a camera. Without it, light rays would be reflected in all directions within the eyeball and would cause diffuse lighting of the retina rather than the normal contrast between dark and light spots required for formation of precise images. The importance of melanin in the pigment layer is well illustrated by its absence in albinos, people who are hereditarily lacking in melanin pigment in all parts of their bodies. When an albino enters a bright room, light that impinges on the retina is reflected in all directions inside the eyeball by the unpigmented surfaces of the retina and by the underlying sclera, so that a single discrete spot of light that would normally excite only a few rods or cones is reflected everywhere and excites many receptors. Therefore, the visual acuity of albinos, even with the best optical correction, is seldom better than 20/100 to 20/200 rather than the normal 20/20 values. The pigment layer also stores large quantities of vitamin A. This vitamin A is exchanged back and forth through the cell membranes of the outer segments of the rods and cones, which themselves are embedded in the pigment. Vitamin A is an important precursor of the photosensitive chemicals of the rods and cones. And now, you have to answer the question and write in the chat. I will look and note in the electronic journal who is present at the lecture. If there is no answer, then I will post absence. Retinal detachment. The neural retina occasionally detaches from the pigment epithelium. In some instances, the cause of such detachment is injury to the eyeball that allows fluid or blood to collect between the neural retina and the pigment epithelium. Detachment is occasionally caused by contracture of fine collagenous fibrils in the vitreous humor, which pull areas of the retina toward the interior of the globe. Partly because of diffusion across the detachment gap and partly because of the independent blood supply to the neural retina through the retinal artery, the detached retina can resist degeneration for days and can become functional again if it is surgically replaced in its normal relation with the pigment epithelium. If it is not replaced soon, however, the retina will be destroyed and will be unable to function even after surgical repair. And now, you have to answer the question and write in the chat. I will look and note in the electronic journal who is present at the lecture. If there is no answer, then I will post absence. Blood supply of the retina, the central retinal artery and the choroid. The nutrient blood supply for the internal layers of the retina is derived from the central retinal artery, which enters the eyeball through the center of the optic nerve and then divides to supply the entire inside retinal surface. Thus, the inner layers of the retina have their own blood supply independent of the other structures of the eye. However, the outermost layer of the retina is adherent to the choroid, which is also a highly vascular tissue lying between the retina and the sclera. The outer layers of the retina, especially the outer segments of the rods and cones, depend mainly on diffusion from the choroid blood vessels for their nutrition, especially for their oxygen. Photochemistry of vision. Both rods and cones contain chemicals that decompose on exposure to light and, in the process, excite the nerve fibers leading from the eye. The light-sensitive chemical in the rods is called rhodopsin. The light-sensitive chemicals in the cones, called cone pigments or color pigments, have compositions only slightly different from that of rhodopsin. In this section, we discuss principally the photochemistry of rhodopsin, but the same principles can be applied to the cone pigments. Rhodopsin retinal visual cycle, and excitation of the rods. Rhodopsin and its decomposition by light energy. The outer segment of the rod that projects into the pigment layer of the retina has a concentration of about 40% of the light-sensitive pigment called rhodopsin, or visual purple. This substance is a combination of the protein scotopsin and the carotenoid pigment retinal, also called retinine. Furthermore, the retinal is a particular type called 11-cis retinal. This cis form of retinal is important because only this form can bind with scotopsin to synthesize rhodopsin. When light energy is absorbed by rhodopsin, the rhodopsin begins to decompose within a very small fraction of a second, as shown at the top of figure. The cause of this is photoactivation of electrons in the retinal portion of the rhodopsin, which leads to instantaneous change of the cis form of retinal into an all-trans form that still has the same chemical structure as the cis form but has a different physical structure, a straight molecule rather than an ungulated molecule. Because the three-dimensional orientation of the reactive sites of the all-trans retinal no longer fits with the orientation of the reactive sites on the protein scotopsin, the all-trans retinal begins to pull away from the scotopsin. Figure. Rhodopsin retinal visual cycle in the rod, showing decomposition of rhodopsin during exposure to light and subsequent slow reformation of rhodopsin by the chemical processes. The immediate product is bathorrhodopsin, which is a partially split combination of the all-trans retinal and scotopsin. Bathorrhodopsin is extremely unstable and decays in nanoseconds to lemurrhodopsin. This then decays in microseconds to metorrhodopsin I, then in about a millisecond to metorrhodopsin II, and finally, much more slowly, in seconds, into the completely split products scotopsin and all-trans retinal. It is the metorrhodopsin II, also called activated rhodopsin, that excites electrical changes in the rods, and the rods then transmit the visual image into the central nervous system in the form of optic nerve action potential, as we discuss later. Reformation of rhodopsin. The first stage in reformation of rhodopsin, as shown in figure, is to reconvert the all-trans retinal into 11-cis retinal. This process requires metabolic energy and is catalyzed by the enzyme retinal isomerase. Once the 11-cis retinal is formed, it automatically recombines with the scotopsin to reform rhodopsin, which then remains stable until its decomposition is again triggered by absorption of light energy. Role of vitamin A for formation of rhodopsin. Note in figure that there is a second chemical route by which all-trans-retinol can be converted into 11-cis-retinol. This is by conversion of the all-trans-retinol first into all-trans-retinol, which is one form of vitamin A. Then the all-trans-retinol is converted into 11-cis-retinol under the influence of the enzyme isomerase. Finally, the 11-cis-retinol is converted into 11-cis-retinol, which combines with scotopsin to form new rhodopsin. Vitamin A is present both in the cytoplasm of the rods and in the pigment layer of the retina. Therefore, vitamin A is normally always available to form new retinol when needed. Conversely, when there is excess retinol in the retina, it is converted back into vitamin A, thus reducing the amount of light-sensitive pigment in the retina. We shall see later that this interconversion between retinol and vitamin A is especially important in long-term adaptation of the retina to different light intensities.
You're right, ma'am. Carrots did give me good eyesight. Now I can spot vegetables I don't like a mile away.
Night blindness. Night blindness occurs in any person with severe vitamin A deficiency. The simple reason for this is that without vitamin A, the amounts of retinol and rhodopsin that can be formed are severely depressed. This condition is called night blindness because the amount of light available at night is too little to permit adequate vision in vitamin A deficient persons. For night blindness to occur, a person usually must remain on a vitamin A deficient diet for months, because large quantities of vitamin A are normally stored in the liver and can be made available to the eyes. Once night blindness develops, it can sometimes be reversed in less than 1 hour by intravenous injection of vitamin A. The literal translation from Russian night blindness is chicken blindness or blind chicken. Excitation of the rod when rhodopsin is activated by light. The rod receptor potential is hyperpolarizing, not depolarizing. When the rod is exposed to light, the resulting receptor potential is different from the receptor potentials in almost all other sensory receptors. That is, excitation of the rod causes increased negativity of the intra-rod membrane potential, which is a state of hyperpolarization, meaning that there is more negativity than normal inside the rod membrane. This is exactly opposite to the decreased negativity, the process of depolarization, that occurs in almost all other sensory receptors. But how does activation of rhodopsin cause hyperpolarization? The answer is that when rhodopsin decomposes, it decreases the rod membrane conductance for sodium ions in the outer segment of the rod. This causes hyperpolarization of the entire rod membrane in the following way. In the dark figure shows movement of potassium ions in a complete electrical circuit through the leaked channels of the inner segments of the rod, thereby creating a negative potential on the inside of the entire cell. However, the outer segment of the rod, where the photoreceptor discs are located, is entirely different. Here, the rod membrane, in the dark state, is very leaky to sodium ions. Therefore, positively charged sodium ions continually leak back to the inside of the rod and thereby neutralize much of the negativity on the inside of the entire cell. Thus, under normal dark conditions, when the rod is not excited, there is reduced electronegativity inside the membrane of the rod, measuring about 40 MV rather than the usual 70 to 80 MV found in most sensory receptors. Then, when the rhodopsin in the outer segment of the rod is exposed to light, the rhodopsin begins to decompose, and this decreases the outer segment membrane sodium conductance of sodium to the interior of the rod. Potassium ions continue to escape through the membrane of the inner segment. Because potassium ions are positive ions, their loss from inside the rod creates increased negativity inside the membrane, and the greater the amount of light energy striking the rod, the greater the electronegativity becomes, that is, the greater is the degree of hyperpolarization. At maximum light intensity, the membrane potential approaches 70 to 80 MV, which is near the equilibrium potential for potassium ions across the membrane. There is a significant difference in the explanation of hyperpolarization of rods and cones of the retina under the action of light in Guyton's textbook and in our lecture. Guyton explains this phenomenon by pumping sodium out of the cell, using sodium-potassium ATPase, probably. See diagram on the left. We explain this phenomenon by the release of potassium from the cell through leakage channels, passive transport. See diagram on the right. This explanation is generally accepted today. Duration of the receptor potential, and logarithmic relation of the receptor potential to light intensity. When a sudden pulse of light strikes the retina, the transient hyperpolarization that occurs in the rods, that is, the receptor potential that occurs, reaches a peak in about 0.3 seconds and lasts for more than a second. In cones, the change occurs 4 times as fast as in the rods. A visual image impinged on the rods of the retina for only 1/millionth of a second can sometimes cause the sensation of seeing the image for longer than a second. Another characteristic of the receptor potential is that it is approximately proportional to the logarithm of the light intensity. This is exceedingly important, because it allows the eye to discriminate light intensities through a range many thousand times as great as would be possible otherwise. To understand the photoreceptor's behavior to light intensities, it is necessary to understand the roles of different currents. There is an ongoing outward potassium current through non-gated K+ selective channels. This outward current tends to hyperpolarize the photoreceptor at around 70 MV, the equilibrium potential for K+. There is also an inward sodium current carried by CGMP-gated sodium channels. This dark current depolarizes the cell to around 40 MV. This is significantly more depolarized than most other neurons. A high density of Na plus K and pumps enables the photoreceptor to maintain a steady intracellular concentration of Na and Nk+. And now, you have to answer the question and write in the chat. I will look and note in the electronic journal who is present at the lecture. If there is no answer, then I will post absence. Representation of molecular steps in photoactivation, modified from Liskov et al., 2000. Depicted as an outer membrane disk in a rod. Step 1, incident photon, H nu, is absorbed and activates a rhodopsin by conformational change in the disk membrane to R star. Step 2, next, R asterisk makes repeated contacts with transducin molecules, catalyzing its activation to G asterisk by the release of bound GDP in exchange for cytoplasmic GTP, which expels its beta and gamma subunits. Step 3, G asterisk binds inhibitory gamma subunits of the phosphodiesterase, PDE, activating its alpha and beta subunits. Step 4, activated PDE hydrolyzes CGMP. Step 5, guanylyl cyclase, GC, synthesizes CGMP, the second messenger in the phototransduction cascade. Reduced levels of cytosolic CGMP cause cyclic nucleotide-gated channels to close, preventing further influx of Na and NCA2+. Representation of the molecular stages of photoactivation in a different form. Presentation of molecular photoactivation step by step. Step 1. A light photon interacts with a retinal molecule in an opsin complex in a photoreceptor cell. The retinal undergoes isomerization, changing from the 11-cis retinal to the all-trans retinal configuration. Opsin therefore undergoes a conformational change to metarhodopsin 2. Presentation of molecular photoactivation step by step. Step 2. Metarhodopsin 2 activates a G protein known as transducin. This causes transducin to dissociate from its bound GDP, and bind GTP, then the alpha subunit of transducin dissociates from the beta and gamma subunits, with the GTP still bound to the alpha subunit. Step 3. The alpha subunit GTP complex activates phosphodiesterase, also known as PDE6. It binds to one of two regulatory subunits of PDE, which itself is a tetramer, and stimulates its activity. Step 4. PDE hydrolyzes CGMP, forming GMP. This lowers the intracellular concentration of CGMP and therefore the sodium channels close. Deactivation of the phototransduction cascade. In light, low CGMP levels close Na and NCA2+ channels, reducing intracellular Na and NCA2+. During recovery, dark adaptation, the low Ca2+ levels induce recovery, termination of the phototransduction cascade, as follows. 1. Low intracellular Ca2+ causes Ca2+ to dissociate from guanylate cyclase activating protein, GCAP. The liberated GCAP ultimately restores depleted CGMP levels, which reopens the cGMP-gated cation channels, restoring dark current. Too low intracellular Ca2+ causes Ca2+ to dissociate from GPAS-activating protein, GAP, also known as regulator of G protein signaling. The liberated GAP deactivates transducin, terminating the phototransduction cascade, restoring dark current. 3 low intracellular Ca2+ makes intracellular Ca recoverin RK dissociate into Ca2+ and recoverin and rhodopsin kinase, RK. The liberated RK then phosphorylates the metrrhodopsin 2, reducing its binding affinity for transducin. Arrestin then completely deactivates the phosphorylated metrrhodopsin 2, terminating the phototransduction cascade, restoring dark current. 4 low intracellular Ca2+ make the Ca2+/colmodulin complex within the cGMP-gated cation channels more sensitive to low cGMP levels, thereby keeping the cGMP-gated cation channel open even at low cGMP levels, restoring dark current. In more detail, G.T.P.ASE accelerating protein, G.A.P, of RGS, regulators of G protein signaling, interacts with the alpha subunit of transducin, and causes it to hydrolyse its bound GTP to GDP, and thus halts the action of phosphodiesterase, stopping the transformation of cGMP to GMP. This deactivation step of the phototransduction cascade, the deactivation of the G protein transducer, was found to be the rate-limiting step in the deactivation of the phototransduction cascade. In other words, guanylate cyclase activating protein, GCAP, is a calcium-binding protein, and as the calcium levels in the cell have decreased, GCAP dissociates from its bound calcium ions, and interacts with guanylate cyclase, activating it. Guanylate cyclase then proceeds to transform GTP to C.G.M.P, replenishing the cell's cGMP levels and thus reopening the sodium channels that were closed during phototransduction. Finally, metrrhodopsin 2 is deactivated. Recoverin, another calcium-binding protein, is normally bound to rhodopsin kinase when calcium is present. When the calcium levels fall during phototransduction, the calcium dissociates from recoverin, and rhodopsin kinase is released and phosphorylates metrrhodopsin 2, which decreases its affinity for transducin. Finally, arrestin, another protein, binds the phosphorylated metrrhodopsin 2, completely deactivating it. Thus, finally, phototransduction is deactivated, and the dark current and glutamate release is restored. It is this pathway, where metrrhodopsin 2 is phosphorylated and bound to arrestin and thus deactivated, which is thought to be responsible for the S2 component of dark adaptation. The S2 component represents a linear section of the dark adaptation function present at the beginning of dark adaptation for all bleaching intensities. Photochemistry of color vision by the cones. The photochemicals in the cones have almost exactly the same chemical composition as that of rhodopsin in the rods. The only difference is that the protein portions, or the opsins called photopsins in the cones, are slightly different from the scotopsin of the rods. The retinal portion of all the visual pigments is exactly the same in the cones as in the rods. The color-sensitive pigments of the cones, therefore, are combinations of retinal and photopsins. In the discussion of color vision later in the lecture, it will become evident that only one of 3 types of color pigments is present in each of the different cones, thus making the cones selectively sensitive to different colors, blue, green, or red. These color pigments are called, respectively, blue-sensitive pigment, green-sensitive pigment, and red-sensitive pigment. The absorption characteristics of the pigments in the 3 types of cones show peak absorbencies at light wavelengths of 445, 535, and 570 nanometers, respectively. These are also the wavelengths for peak light sensitivity for each type of cone, which begins to explain how the retina differentiates the colors. The approximate absorption curves for these 3 pigments are shown in figure. Also shown is the absorption curve for the rhodopsin of the rods, with a peak at 505 nanometers. Figure. Light absorption by the pigment of the rods and by the pigments of the 3 color-receptive cones of the human retina. Automatic regulation of retinal sensitivity. Light and dark adaptation. Light and dark adaptation. If a person has been in bright light for hours, large portions of the photochemicals in both the rods and the cones will have been reduced to retinal and opsins. Furthermore, much of the retinal of both the rods and the cones will have been converted into vitamin A. Because of these 2 effects, the concentrations of the photosensitive chemicals remaining in the rods and cones are considerably reduced, and the sensitivity of the eye to light is correspondingly reduced. This is called light adaptation. Conversely, if a person remains in darkness for a long time, the retinal and opsins in the rods and cones are converted back into the light-sensitive pigments. Furthermore, vitamin A is converted back into retinal to give still more light-sensitive pigments, the final limit being determined by the amount of opsins in the rods and cones to combine with the retinal. This is called dark adaptation. Figure shows the course of dark adaptation when a person is exposed to total darkness after having been exposed to bright light for several hours. Note that the sensitivity of the retina is very low on first entering the darkness, but within 1 minute, the sensitivity has already increased tenfold, that is, the retina can respond to light of 1/10 the previously required intensity. At the end of 20 minutes, the sensitivity has increased about 6,000-fold, and at the end of 40 minutes, about 25,000-fold. Figure. Dark adaptation, demonstrating the relation of cone adaptation to rod adaptation. The resulting curve of figure is called the dark adaptation curve. Note, however, the inflection in the curve. The early portion of the curve is caused by adaptation of the cones, because all the chemical events of vision, including adaptation, occur about 4 times as rapidly in cones as in rods. However, the cones do not achieve anywhere near the same degree of sensitivity change in darkness as the rods do. Therefore, despite rapid adaptation, the cones cease adapting after only a few minutes, while the slowly adapting rods continue to adapt for many minutes and even hours, their sensitivity increasing tremendously. In addition, still more sensitivity of the rods is caused by neuronal signal convergence of 100 or more rods onto a single ganglion cell in the retina. These rods summit to increase their sensitivity, as discussed later in the chapter. Other mechanisms of light and dark adaptation. In addition to adaptation caused by changes in concentrations of rhodopsin or color photochemicals, the eye has 2 other mechanisms for light and dark adaptation. The first of these is a change in pupillary size. This can cause adaptation of approximately 30-fold within a fraction of a second, because of changes in the amount of light allowed through the pupillary opening. The other mechanism is neural adaptation, involving the neurons in the successive stages of the visual chain in the retina itself and in the brain. That is, when light intensity first increases, the signals transmitted by the bipolar cells, horizontal cells, amacrine cells, and ganglion cells are all intense. However, most of these signals decrease rapidly at different stages of transmission in the neural circuit. Although the degree of adaptation is only a few-fold rather than the many thousand-fold that occurs during adaptation of the photochemical system, neural adaptation occurs in a fraction of a second, in contrast to the many minutes to hours required for full adaptation by the photochemicals. Value of light and dark adaptation in vision. Between the limits of maximal dark adaptation and maximal light adaptation, the eye can change its sensitivity to light as much as 500,000 to 1 million times, the sensitivity automatically adjusting to changes in illumination. Because registration of images by the retina requires detection of both dark and light spots in the image, it is essential that the sensitivity of the retina always be adjusted so that the receptors respond to the lighter areas but not to the darker areas. An example of maladjustment of retinal adaptation occurs when a person leaves a movie theater and enters the bright sunlight. Then, even the dark spots in the images seem exceedingly bright, and as a consequence, the entire visual image is bleached, having little contrast among its different parts. This is poor vision, and it remains poor until the retina has adapted sufficiently so that the darker areas of the image no longer stimulate the receptors excessively. Conversely, when a person first enters darkness, the sensitivity of the retina is usually so slight that even the light spots in the image cannot excite the retina. After dark adaptation, the light spots begin to register. As an example of the extremes of light and dark adaptation, the intensity of sunlight is about 10 billion times that of starlight, yet the eye can function both in bright sunlight after light adaptation and in starlight after dark adaptation. Color vision. The different cones are sensitive to different colors of light. This section is a discussion of the mechanisms by which the retina detects the different gradations of color in the visual spectrum. Color vision. Color vision is found in many animals and is mediated by similar underlying mechanisms with common types of biological molecules and a complex history of evolution in different animal taxa. In primates, Color vision may have evolved under selective pressure for a variety of visual tasks, including the foraging for nutritious young leaves, ripe fruit, and flowers, as well as detecting predator camouflage and emotional states in other primates. Color vision, a feature of visual perception, is an ability to perceive differences between light composed of different frequencies independently of light intensity. Color perception is a part of the larger visual system and is mediated by a complex process between neurons that begins with differential stimulation of different types of photoreceptors by light entering the eye. Those photoreceptors then emit outputs that are propagated through many layers of neurons and then ultimately to the brain. Wavelength. Isaac Newton discovered that white light, after being split into its component colors when passed through a dispersive prism, could be recombined to make white light by passing them through a different prism. The visible light spectrum ranges from about 380 to 740 nanometers. Spectral colors, colors that are produced by a narrow band of wavelengths, such as red, orange, yellow, green, cyan, blue, and violet, can be found in this range. These spectral colors do not refer to a single wavelength, but rather to a set of wavelengths: red, 625 to 740 nanometers, orange, 590 to 625 nanometers, yellow, 565 to 590 nanometers, green, 500 to 565 nanometers, cyan, 485 to 500 nanometers, blue, 450 to 485 nanometers, violet, 380 to 450 nanometers. Wavelengths longer or shorter than this range are called infrared or ultraviolet, respectively. Humans cannot generally see these wavelengths, but other animals may. Dimensionality. Color vision is categorized foremost according to the dimensionality of the color gamut, which is defined by the number of primaries required to represent the color vision. This is generally equal to the number of photopsins expressed, a correlation that holds for vertebrates but not invertebrates. The common vertebrate ancestor possessed 4 photopsins, expressed in cones, plus rhodopsin, expressed in rods, so was tetrachromatic. However, many vertebrate lineages have lost one or many photopsin genes, leading to lower dimension color vision. The dimensions of color vision range from 1-dimensional and up. Monochromacy, 1D color vision, lack of any color perception. Dichromacy, 2D color vision, dimensionality of most mammals and a quarter of color-blind humans. Trichromacy, 3D color vision, dimensionality of most humans. Tetrachromacy, 4D color vision, dimensionality of most birds, reptiles, and fish. Pentachromacy and higher, 5D and color vision, rare invertebrates. Physiology of color perception. Perception of color begins with specialized retinal cells known as cone cells. Cone cells contain different forms of opsin, a pigment protein, that have different spectral sensitivities. Humans contain 3 types, resulting in trichromatic color vision. Each individual cone contains pigments composed of opsin apoprotein covalently linked to a light-absorbing prosthetic group, either 11-cis-hydroretinal or, more rarely, 11-cis-dehydroretinal. The cones are conventionally labeled according to the ordering of the wavelengths of the peaks of their spectral sensitivities: short, S, medium, M, and long, L, cone types. These 3 types do not correspond well to particular colors as we know them. Rather, the perception of color is achieved by a complex process that starts with the differential output of these cells in the retina and which is finalized in the visual cortex and associative areas of the brain. Normalized response spectra of human cones to monochromatic spectral stimuli, with wavelength given in nanometers. For example, while the L cones have been referred to simply as red receptors, microspectrophotometry has shown that their peak sensitivity is in the greenish-yellow region of the spectrum. Similarly, the S cones and M cones do not directly correspond to blue and green, although they are often described as such. The RGB color model, therefore, is a convenient means for representing color but is not directly based on the types of cones in the human eye. The peak response of human cone cells varies, even among individuals with so-called normal color vision. In some non-human species this polymorphic variation is even greater, and it may well be adaptive. Theories of color vision. 2 complementary theories of color vision are the trichromatic theory and the opponent process theory. Authors of 2 major color vision theories, Jung-Helmholtz trichromatic theory, left, and Herring's opposite process theory, right. Tricolor mechanism of color detection. All theories of color vision are based on the well-known observation that the human eye can detect almost all gradations of colors when only red, green, and blue monochromatic lights are appropriately mixed in different combinations. The trichromatic theory, or Young-Helmholtz theory, proposed in the 19th century by Thomas Young and Hermann von Helmholtz, posits three types of cones preferentially sensitive to blue, green, and red, respectively. Others have suggested that the trichromatic theory is not specifically a theory of color vision but a theory of receptors for all vision, including color but not specific or limited to it. Equally, it has been suggested that the relationship between the phenomenal opponency described by Hering and the physiological opponent processes are not straightforward, making of physiological opponency a mechanism that is relevant to the whole of vision, and not just to color vision alone. Spectral sensitivities of the three types of cones. On the basis of color vision tests, the spectral sensitivities of the three types of cones in humans have proved to be essentially the same as the light absorption curves for the three types of pigment found in the cones. These curves are shown in Figure A and slightly differently in Figure B. They can explain most of the phenomena of color vision. Spectral sensitivities of the three types of cones. Figure A. Light absorption by the pigment of the rods and by the pigments of the three color receptive cones of the human retina. Figure B. Demonstration of the degree of stimulation of the different color-sensitive cones by monochromatic lights of four colors: blue, green, yellow, and orange. Interpretation of color in the nervous system. Referring to Figure, one can see that an orange monochromatic light with a wavelength of 580 nanometers stimulates the red cones to a stimulus value of about 99% of the peak stimulation at optimum wavelength. It stimulates the green cones to a stimulus value of about 42, but the blue cones not at all. Thus, the ratios of stimulation of the three types of cones in this instance are 99:42:0. The nervous system interprets this set of ratios as the sensation of orange. Conversely, a monochromatic blue light with a wavelength of 450 nanometers stimulates the red cones to a stimulus value of 0, the green cones to a value of 0, and the blue cones to a value of 97. This set of ratios 0:0:97 is interpreted by the nervous system as blue. Likewise, ratios of 83:83:0 are interpreted as yellow, and 31:67:36 as green. Perception of white light. About equal stimulation of all the red, green, and blue cones gives one the sensation of seeing white. Yet there is no single wavelength of light corresponding to white. Instead, white is a combination of all the wavelengths of the spectrum. Furthermore, the perception of white can be achieved by stimulating the retina with a proper combination of only three chosen colors that stimulate the respective types of cones about equally. The failure of trichromatic theory to account for afterimages indicates the need for an opponent process theory such as that articulated by Ewald Hering, 1878, and further developed by Hervik and Jameson, 1957. Afterimage. An afterimage is an image that continues to appear in the eyes after a period of exposure to the original image. An afterimage may be a normal phenomenon, physiological afterimage, or may be pathological, palinopsia. Illusory palinopsia may be a pathological exaggeration of physiological afterimages. Afterimages occur because photochemical activity in the retina continues even when the eyes are no longer experiencing the original stimulus. Complementary colors. When staring at a bright color for a while, e.g. red, then looking away at a white field, an afterimage is perceived, such that the original color will evoke its complementary color, green, in the case of red input. When complementary colors are combined or mixed, they cancel each other out and become neutral, white or gray. That is, complementary colors are never perceived as a mixture. There is no greenish red or yellowish blue, despite claims to the contrary. The strongest color contrast a color can have is its complementary color. Complementary colors may also be called opposite colors and are understandably the basis of the colors used in the opponent process theory. Opponent color pairs based on the NCS experiment, including black, white and the four unique hues. And now, you have to answer the question and write in the chat. I will look and note in the electronic journal who is present at the lecture. If there is no answer, then I will post absence. Look at the dot plus in the middle of the blue figure above for 15 to 30 seconds. Then look at the tiny dot plus in the center of the white square on the right. The reversal of the color you see is an example of an after image. Notice how long the after image continues before it disappears. Write in the general chat, what color did the cross and background invert their colors to? Look at the middle of the figure above for 15 to 30 seconds. Then look at the center of the white square on the right. What color inversion do you see? Write in the general chat, what color did the cross and background invert their colors to? The lilac chaser is a visual illusion, also known as the Pachmann illusion. It consists of 12 lilac, or pink, rose, or magenta, blurred discs arranged in a circle, like the numbers on a clock, around a small black, central cross on a gray background. One of the discs disappears briefly, for about 0.1 seconds, then the next, about 0.125 seconds later, and the next, and so on, in a clockwise direction. When one stares at the cross for at least 30 seconds, one sees three illusions. A gap running around the circle of lilac discs. A green disc running around the circle of lilac discs in place of the gap, and the green disc running around on the gray background, with the lilac discs having disappeared in sequence. The illusion was created by Jeremy Hinton sometime before 2005. It then spread widely over the internet. It has been praised for being a striking visual illusion, and has been the subject of academic research. Look at the cross for at least 30 seconds. What color disc replaces the gap running in a circle of blue discs? Negative after image in the eye by closed eyes. If you gaze a colored image with open eyes for about 30 s and you look at white screen with your open eyes, you can see a negative after image that is the same size and shape but with complementary colors. Nevertheless, you can see a negative after image not only by means of open eyes but also by closed eyes without any ambient photonic stimulation. See the simple illustration. The photopigment bleaching hypothesis, Rushton and Henry, 1968, Williams and MacLeod, 1979, cannot give explanation for the fact that if your eyes are closed nevertheless a long-lasting signal is generated that causes a static negative after image inside your eyes interpreted then by neural mechanisms within the brain. Opponent process. The opponent process is a color theory that states that the human visual system interprets information about color by processing signals from photoreceptor cells in an antagonistic manner. The opponent process theory suggests that there are three opponent channels, each comprising an opposing color pair, red versus green, blue versus yellow, and black versus white, luminance. The theory was first proposed in 1892 by the German physiologist Ewald Hering. Physiological basis. Relation to LMS color space. Though the trichromatic and opponent processes theories were initially thought to be at odds, it was later shown that the mechanisms responsible for the opponent process receive signals from the three types of cones predicted by the trichromatic theory and process them at a more complex level. Most humans have three different cone cells in their retinas that facilitate trichromatic color vision. Colors are determined by the proportional excitation of these three cone types, i.e. their quantum catch. The levels of excitation of each cone type are the parameters that define LMS color space. To calculate the opponent process tristimulus values from the LMS color space, the cone excitations must be compared. The luminous opponent channel is equal to the sum of all three cone cells, plus the rod cells in some conditions. The red, green opponent channel is equal to the difference of the L and M cones. The blue, yellow opponent channel is equal to the difference of the S cone and the sum of the L and M cones. Let's say it again. Diagram of the opponent process. To calculate the opponent process tristimulus values from the LMS color space, the cone excitations must be compared. The luminous opponent channel is equal to the sum of all three cone cells, plus the rod cells in some conditions. The red, green opponent channel is equal to the difference of the L and M cones. The blue, yellow opponent channel is equal to the difference of the S cone and the sum of the L and M cones. Color blindness. Color blindness. Red-green color blindness. When a single group of color-receptive cones is missing from the eye, the person is unable to distinguish some colors from others. For instance, one can see in figure that green, yellow, orange, and red colors, which are the colors between the wavelengths of 525 and 675 nanometers, are normally distinguished from one another by the red and green cones. If either of these two cones is missing, the person cannot use this mechanism for distinguishing these four colors. The person is especially unable to distinguish red from green and is therefore said to have red-green color blindness. A person with loss of red cones is called a protanope. The overall visual spectrum is noticeably shortened at the long wavelength end because of a lack of the red cones. A colorblind person who lacks green cones is called a deuteranope. This person has a perfectly normal visual spectral width because red cones are available to detect the long wavelength red color. Red-green color blindness is a genetic disorder that occurs almost exclusively in males. That is, genes in the female X chromosome code for the respective cones. Yet color blindness almost never occurs in females because at least one of the two X chromosomes almost always has a normal gene for each type of cone. Because the male has only one X chromosome, a missing gene can lead to color blindness. Because the X chromosome in the male is always inherited from the mother, never from the father, color blindness is passed from mother to son, and the mother is said to be a color blindness carrier. This is true in about 8% of all women. Blue weakness. Only rarely are blue cones missing, although sometimes they are underrepresented, which is a genetically inherited state giving rise to the phenomenon called blue weakness. Can animals see color? It was once thought that animals, including cats and dogs, could only see in black and white. However, scientists have proven this to be a myth. In all animals, including humans, the perception of color is determined by the presence of cells in the eye called cone photoreceptors. Cats and dogs are colorblind. Cats and dogs only have blue and green cones. This means they have a much more muted perception of color, which is akin to color blindness in humans. As cats and dogs are not sensitive to red light, they have difficulty distinguishing some colors. For example, behavioral tests in dogs suggest that they can distinguish red from blue, but often confuse red and green. Similarly, they often perceive green as gray. Only one animal cannot see in color. The only animal that has been confirmed to see only in black and white is a fish called a skate. This is because it has no cones in its eyes. Color test charts. A rapid method for determining color blindness is based on the use of spot charts such as those shown in figure. These charts are arranged with a confusion of spots of several different colors. In the top chart, the person with normal color vision reads 74, whereas the red-green colorblind person reads 21. In the bottom chart, the person with normal color vision reads 42, whereas the red-blind person reads 2, and the green-blind person reads 4. If one studies these charts while at the same time observing the spectral sensitivity curves of the different cones depicted in figure, it can be readily understood how excessive emphasis can be placed on spots of certain colors by colorblind people. The Ishihara test is a color vision test for detection of red-green color deficiencies. It was named after its designer, Shinobu Ishihara, a professor at the University of Tokyo, who first published his tests in 1917. Ishihara had just completed his graduate studies in ophthalmology in Germany when war broke out in Europe and World War I had begun. While holding a military position related to his field, he was given the task of creating a color blindness test. Ishihara studied existing tests and combined elements of the Stilling test, named after the German ophthalmologist Jacob Stilling, with the concept of pseudo-isochromaticism to produce an improved, more accurate and easier to use test. Color vision test. The Ishihara test consists of a number of Ishihara plates, which are a type of pseudo-isochromatic plate. Each plate depicts a solid circle of colored dots appearing randomized in color and size. Within the pattern are dots which form a number or shape clearly visible to those with normal color vision, and invisible, or difficult to see, to those with a red-green color vision defect. Other plates are intentionally designed to reveal numbers only to those with a red-green color vision deficiency, and be invisible to those with normal red-green color vision. The full test consists of 38 plates, but the existence of a severe deficiency is usually apparent after only a few plates. There are also Ishihara tests consisting of 10, 14, or 24 test plates, and plates in some versions ask the viewer to trace a line rather than read a number. Let's test you for color blindness. Be sure to answer. The quality of the answer is not evaluated. What can you see in these pictures? Tip: point of view matters. Yes, there are butterflies. Red-green color blindness is the most common color deficiency type. By the way, it affects men much more often than women. What is hidden in all of these dots? Do you see 7 and 16? How about some more numbers? It's 13 and 8. People with red color blindness would see 1, and with green color blindness 3, and both of them would see 3 instead of 8. Let's make it a little harder. Did you find 12 and 9? People with red-green color blindness see 17 and 0. Does it look simple? See something? Try one more time. Still nothing? That's right. Yet, people with red-green color blindness see 45. What comes out of the blue? No, it's not the pool floor. 503 is hiding here. Do you recognize the green screen? Action. No, we're not going to shoot anything, just finding the number 276. This rave of red hides some numbers. Vision test, check how good your eyes are. Yes, red colors can be very different. Some people can't see similar shades among the others. How about you? Can you see 641? If you gaze into the abyss, what do you see? Do you expect the little mermaid to swim out of the deep? No, she sent her friend 84 to tell you that your eyes are perfect. Can you read the whole line, or sing it? I am colorblind, coffee black and egg white. Do you remember this Counting Crows song? And the last question, do you see the word? Daltonism. If you do, you certainly don't have it. We hope you liked our test and recognized everything in the images. But remember, if you have the symptoms mentioned here, consult your eye doctor. So what color is the dress? Black and blue, white, and gold or other colors? As you can see, the dress was a viral phenomenon on the internet in 2015. Viewers of the image disagreed on whether the dress depicted in a photograph was colored black and blue, or white and gold. The phenomenon revealed differences in human color perception, which have been the subject of ongoing scientific investigations into neuroscience and vision science, producing a number of papers published in peer-reviewed scientific journals. Colorblind glasses. Colorblind glasses or color-correcting lenses are light filters, usually in the form of glasses or contact lenses, that attempt to alleviate color blindness by bringing deficient color vision closer to normal color vision or to make certain color tasks easier to accomplish. Despite viral status, the academic literature is generally skeptical of the efficacy of color-correcting lenses. Smart glasses. Google Glass has been used with daltonization filters to create a sort of active color-correcting lens. The color-correcting lenses discussed above are all passive filters, and can therefore only subtract light at certain wavelengths. However, active lenses, which are also able to amplify light at certain wavelengths, are much more flexible in how they can correct color vision and impose bigger shifts of color. Smart glasses like the Google Glass and Epson Moverio can act like active lenses and have been used with recoloring apps to help the colorblind with color tasks. Digital recoloring filters are usually based on daltonization algorithms that recolor the image regardless of the content, but smart glasses can also be context-aware and adapt to different scenes to optimize the filter. For example, they could increase the contrast between brown and pink when specifically cooking red meat. These active lenses are a type of augmented reality. Neural function of the retina. Neural function of the retina. Neural circuitry of the retina. Figure shows the tremendous complexity of neural organization in the retina. Neural circuitry of the retina. The different neuronal cell types are as follows. 1. The photoreceptors themselves, the rods and cones which transmit signals to the outer plexiform layer, where they synapse with bipolar cells and horizontal cells. 2. The horizontal cells, which transmit signals horizontally in the outer plexiform layer from the rods and cones to bipolar cells. 3. The bipolar cells, which transmit signals vertically from the rods, cones, and horizontal cells to the inner plexiform layer, where they synapse with ganglion cells and amacrine cells. For the amacrine cells, which transmit signals in two directions, either directly from bipolar cells to ganglion cells or horizontally within the inner plexiform layer from axons of the bipolar cells to dendrites of the ganglion cells or to other amacrine cells. 5. The ganglion cells, which transmit output signals from the retina through the optic nerve into the brain. A sixth type of neuronal cell in the retina, not very prominent and not shown in the figure, is the interplexiform cell. This cell transmits signals in the retrograde direction from the inner plexiform layer to the outer plexiform layer. These signals are inhibitory and are believed to control lateral spread of visual signals by the horizontal cells in the outer plexiform layer. Their role may be to help control the degree of contrast in the visual image. The visual pathway from the cones to the ganglion cells functions. Neural circuitry of the retina. As is true for many of our other sensory systems, the retina has both an old type of vision based on rod vision and a new type of vision based on cone vision. The neurons and nerve fibers that conduct the visual signals for cone vision are considerably larger than those that conduct the visual signals for rod vision, and the signals are conducted to the brain 2 to 5 times as rapidly. Also, the circuitry for the two systems is slightly different, as follows. The figure is the visual pathway from the foveal portion of the retina, representing the new, fast cone system. This shows 3 neurons in the direct pathway, 1. Cones, 2. Bipolar cells, and 3. Ganglion cells. In addition, horizontal cells transmit inhibitory signals laterally in the outer plexiform layer, and amacrine cells transmit signals laterally in the inner plexiform layer. The figure are the neural connections for the peripheral retina, where both rods and cones are present. 3 bipolar cells are shown. The middle of these connects only to rods, representing the type of visual system present in many lower animals. The output from the bipolar cell passes only to amacrine cells, which relay the signals to the ganglion cells. Thus, for pure rod vision, there are 4 neurons in the direct visual pathway, 1. Rods, 2. Bipolar cells, 3. Amacrine cells, and 4. Ganglion cells. Also, horizontal and amacrine cells provide lateral connectivity. The other 2 bipolar cells shown in the peripheral retinal circuitry of figure connect with both rods and cones. The outputs of these bipolar cells pass both directly to ganglion cells and by way of amacrine cells. Neurotransmitters released by retinal neurons. Not all the neurotransmitter chemical substances used for synaptic transmission in the retina have been entirely delineated. However, both the rods and the cones release glutamate at their synapses with the bipolar cells. Histological and pharmacological studies have shown there to be many types of amacrine cells secreting at least 8 types of transmitter substances, including gamma-aminobutyric acid, glycine, dopamine, acetylcholine, and indolamine, all of which normally function as inhibitory transmitters. The transmitters of the bipolar, horizontal, and interplexiform cells are unclear, but at least some of the horizontal cells release inhibitory transmitters. Transmission of most signals occurs in the retinal neurons by electrotonic conduction, not by action potentials. The only retinal neurons that always transmit visual signals by means of action potentials are the ganglion cells, and they send their signals all the way to the brain through the optic nerve. Occasionally, action potentials have also been recorded in amacrine cells, although the importance of these action potentials is questionable. Otherwise, all the retinal neurons conduct their visual signals by electrotonic conduction, which can be explained as follows. Electrotonic conduction means direct flow of electric current, not action potentials, in the neuronal cytoplasm and nerve axons from the point of excitation all the way to the output synapses. Even in the rods and cones, conduction from their outer segments, where the visual signals are generated, to the synaptic bodies is by electrotonic conduction. That is, when hyperpolarization occurs in response to light in the outer segment of a rod or a cone, almost the same degree of hyperpolarization is conducted by direct electric current flow in the cytoplasm all the way to the synaptic body, and no action potential is required. Then, when the transmitter from a rod or cone stimulates a bipolar cell or horizontal cell, once again the signal is transmitted from the input to the output by direct electric current flow, not by action potentials. The importance of electrotonic conduction is that it allows graded conduction of signal strength. Thus, For the rods and cones, the strength of the hyperpolarizing output signal is directly related to the intensity of illumination. The signal is not all or none, as would be the case for each action potential. However, we know that lateral inhibition occurs at the bipolar cells. Simple math calculations will be involved from here on, so hang tight. We already know that each bipolar cell received a particular response, receptor A generate response of 100 which is sent to bipolar A and so on and so forth. Bipolar cell A, B and C received 100 while bipolar cell D, E, and F received 20. Lateral inhibition to enhance visual contrast function of the horizontal cells. The horizontal cells, shown in figure, connect laterally between the synaptic bodies of the rods and cones, as well as connecting with the dendrites of the bipolar cells. The outputs of the horizontal cells are always inhibitory. Therefore, this lateral connection provides the same phenomenon of lateral inhibition that is important in all other sensory systems, that is, helping to ensure transmission of visual patterns with proper visual contrast. Lateral inhibition. In neurobiology, lateral inhibition is the capacity of an excited neuron to reduce the activity of its neighbors. Lateral inhibition disables the spreading of action potentials from excited neurons to neighboring neurons in the lateral direction. This creates a contrast in stimulation that allows increased sensory perception. It is also referred to as lateral antagonism and occurs primarily in visual processes, but also in tactile, auditory, and even olfactory processing. Cells that utilize lateral inhibition appear primarily in the cerebral cortex and thalamus and make up lateral inhibitory networks, LINs. Artificial lateral inhibition has been incorporated into artificial sensory systems, such as vision chips, hearing systems, and optical mice. An often underappreciated point is that although lateral inhibition is visualized in a spatial sense, it is also thought to exist in what is known as lateral inhibition across abstract dimensions. This refers to lateral inhibition between neurons that are not adjacent in a spatial sense, but in terms of modality of stimulus. This phenomenon is thought to aid in color discrimination. Sensory inhibition. A stimulus affecting all three neurons, but which affects B strongest or first, can be sharpened if B sends lateral signals to neighbors A and C not to fire, thereby inhibiting them. Lateral inhibition is used in vision to sharpen signals to the brain, pink arrow. Visual inhibition. Lateral inhibition increases the contrast and sharpness in visual response. This phenomenon already occurs in the mammalian retina. In the dark, a small light stimulus will enhance the different photoreceptors, rod cells. The rods in the center of the stimulus will transduce the light signal to the brain, whereas different rods on the outside of the stimulus will send a dark signal to the brain due to lateral inhibition from horizontal cells. This contrast between the light and dark creates a sharper image. Compare unsharp masking in digital processing. This mechanism also creates the Mach band visual effect. Mach band visual effect. Along the boundary between adjacent shades of gray in the Mach band's illusion, lateral inhibition makes the darker area falsely appear even darker and the lighter area falsely appear even lighter. Visual lateral inhibition is the process in which photoreceptor cells aid the brain in perceiving contrast within an image. Electromagnetic light enters the eye by passing through the cornea, pupil, and the lens, optics. The rods become stimulated by the energy from the light and release an excitatory neural signal to the horizontal cells. This excitatory signal, however, will only be transmitted by the rod cells in the center of the ganglion cell receptive field to ganglion cells because horizontal cells respond by sending an inhibitory signal to the neighboring rods to create a balance that allows mammals to perceive more vivid images. The central rod will send the light signals directly to bipolar cells which in turn will relay the signal to the ganglion cells. Amacrine cells also produce lateral inhibition to bipolar cells 18 and ganglion cells to perform various visual computations including image sharpening. The final visual signals will be sent to the thalamus and cerebral cortex, where additional lateral inhibition occurs. Marrying lateral inhibition and Mach phenomenon. With all that said, how do we explain the Mach band illusion in terms of lateral inhibition? Papageorgiou and colleague, 1990, as well as Goldstein, 2007, show us how. Imagine that you have 6 photoreceptors, A, B, C, D, E, F. Each receptor sends signals to the bipolar cells, and each bipolar cell sends lateral inhibition to its neighbors. Receptors A, B and C received intense illumination while receptors D, E, and F received dim illumination. Simply put, A, B and C are cast in the light, while D, E, and F are in the shadow. Let's assume that receptors A, B and C generate responses of 100 and receptors D, E, and F generate responses of 20. Without inhibition, these receptors sends the same responses to their bipolar cells. If things stopped here, you would see a bright bar with equal intensity across A, B and C, and a dim bar with equal intensity across D, E, and F. What we need to do now is to determine the amount of inhibition that each bipolar cell receives from its neighbor on each side. For purpose of easy calculation, we will assume that each cell sends 1/10 of the cell's initial output to its neighbors. Thus, cells A, B and C will send 100 x 0.1 equals 10 units of inhibition to their neighbors. Cells D, E, and F will send 20 x 0.1 equals 2 units of inhibition to their neighbors. So now we have to find out the final response of each cell by subtracting the amount of inhibition from the initial response. Remember that each cell receives inhibition from its neighbor on either side. The final calculation. As a result of lateral inhibition, the most significant information is transmitted to the brain. Lateral inhibition to enhance visual contrast function of the horizontal cells. This phenomenon is demonstrated in figure, which shows a minute spot of light focused on the retina. The visual pathway from the centralmost area where the light strikes is excited, whereas an area to the side is inhibited. In other words, instead of the excitatory signal spreading widely in the retina because of spreading dendritic and axonal trees in the plexiform layers, transmission through the horizontal cells puts a stop to this by providing lateral inhibition in the surrounding areas. This is essential to allow high visual accuracy in transmitting contrast borders in the visual image. Some of the amacrine cells probably provide additional lateral inhibition and further enhancement of visual contrast in the inner plexiform layer of the retina as well. Excitation of some bipolar cells and inhibition of others, the depolarizing and hyperpolarizing bipolar cells. Two types of bipolar cells provide opposing excitatory and inhibitory signals in the visual pathway: 1, the depolarizing bipolar cell, and 2, the hyperpolarizing bipolar cell. That is, some bipolar cells depolarize when the rods and cones are excited, and others hyperpolarize. There are two possible explanations for this difference. One explanation is that the two bipolar cells are of entirely different types, one responding by depolarizing in response to the glutamate neurotransmitter released by the rods and cones, and the other responding by hyperpolarizing. The other possibility is that one of the bipolar cells receives direct excitation from the rods and cones, whereas the other receives its signal indirectly through a horizontal cell. Because the horizontal cell is an inhibitory cell, this would reverse the polarity of the electrical response. Regardless of the mechanism for the two types of bipolar responses, the importance of this phenomenon is that it allows half the bipolar cells to transmit positive signals and the other half to transmit negative signals. We shall see later that both positive and negative signals are used in transmitting visual information to the brain. Another important aspect of this reciprocal relation between depolarizing and hyperpolarizing bipolar cells is that it provides a second mechanism for lateral inhibition, in addition to the horizontal cell mechanism. Because depolarizing and hyperpolarizing bipolar cells lie immediately against each other, this provides a mechanism for separating contrast borders in the visual image, even when the border lies exactly between two adjacent photoreceptors. In contrast, the horizontal cell mechanism for lateral inhibition operates over a much greater distance. Amacrine cells and their functions. About 30 types of amacrine cells have been identified by morphological or histochemical means. The functions of about half a dozen types of amacrine cells have been characterized, and all of them are different. One type of amacrine cell is part of the direct pathway for rod vision, that is, from rod to bipolar cells to amacrine cells to ganglion cells. Another type of amacrine cell responds strongly at the onset of a continuing visual signal, but the response dies rapidly. Other amacrine cells respond strongly at the offset of visual signals, but again, the response dies quickly. Still other amacrine cells respond when a light is turned either on or off, signaling simply a change in illumination, irrespective of direction. Still another type of amacrine cell responds to movement of a spot across the retina in a specific direction. Therefore, these amacrine cells are said to be directional sensitive. In a sense, then, many or most amacrine cells are interneurons that help analyze visual signals before they ever leave the retina. Ganglion cells and optic nerve fibers. Each retina contains about 100 million rods and 3 million cones, yet the number of ganglion cells is only about 1.6 million. Thus, an average of 60 rods and 2 cones converge on each ganglion cell and the optic nerve fiber leading from the ganglion cell to the brain. However, major differences exist between the peripheral retina and the central retina. As one approaches the fovea, fewer rods and cones converge on each optic fiber, and the rods and cones also become more slender. These effects progressively increase the acuity of vision in the central retina. In the center, in the central fovea, there are only slender cones, about 35,000 of them, and no rods. Also, the number of optic nerve fibers leading from this part of the retina is almost exactly equal to the number of cones, as shown to the figure. This explains the high degree of visual acuity in the central retina in comparison with the much poorer acuity peripherally. Another difference between the peripheral and central portions of the retina is the much greater sensitivity of the peripheral retina to weak light. This results partly from the fact that rods are 30 to 300 times more sensitive to light than cones are, but it is further magnified by the fact that as many as 200 rods converge on a single optic nerve fiber in the more peripheral portions of the retina, so that signals from the rod's summit to give even more intense stimulation of the peripheral ganglion cells and their optic nerve fibers. 3 types of retinal ganglion cells and their respective fields. There are 3 distinct types of ganglion cells, designated W, X, and Y cells. Each of these serves a different function. Transmission of rod vision by the W cells. The W cells, constituting about 40% of all the ganglion cells, are small, having a diameter less than 10 micrometers, and they transmit signals in their optic nerve fibers at the slow velocity of only 8 m/section. These ganglion cells receive most of their excitation from rods, transmitted by way of small bipolar cells and amacrine cells. They have broad fields in the peripheral retina because the dendrites of the ganglion cells spread widely in the inner plexiform layer, receiving signals from broad areas. On the basis of histology as well as physiologic experiments, the W cells seem to be especially sensitive for detecting directional movement in the field of vision, and they are probably important for much of our crude rod vision under dark conditions. Transmission of the visual image in color by the X cells. The most numerous of the ganglion cells are the X cells, representing 55% of the total. They are of medium diameter, between 10 and 15 micrometers, and transmit signals in their optic nerve fibers at about 14 m/section. The X cells have small fields because their dendrites do not spread widely in the retina. Because of this, their signals represent discrete retinal locations. Therefore, it is mainly through the X cells that the fine details of the visual image are transmitted. Also, because every X cell receives input from at least one cone, X cell transmission is probably responsible for all color vision. Function of the Y cells to transmit instantaneous changes in the visual image. The Y cells are the largest of all, up to 35 micrometers in diameter, and they transmit their signals to the brain at 50 m/sec or faster. They are the least numerous of all the ganglion cells, representing only 5% of the total. Also, they have broad dendritic fields, so that signals are picked up by these cells from widespread retinal areas. The Y ganglion cells respond, like many of the amacrine cells, to rapid changes in the visual image, either rapid movement or rapid change in light intensity, sending bursts of signals for only small fractions of a second. These ganglion cells presumably apprise the central nervous system almost instantaneously when a new visual event occurs anywhere in the visual field, but without specifying with great accuracy the location of the event, other than to give appropriate clues that make the eyes move toward the exciting vision. Excitation of the ganglion cells. Spontaneous, continuous action potentials in the ganglion cells. It is from the ganglion cells that the long fibers of the optic nerve lead into the brain. Because of the distance involved, the electrotonic method of conduction employed in the rods, cones, and bipolar cells within the retina is no longer appropriate. Therefore, ganglion cells transmit their signals by means of repetitive action potentials instead. Furthermore, Even when unstimulated, they still transmit continuous impulses at rates varying between 5 and 40 per second. The visual signals, in turn, are superimposed onto this background ganglion cell firing. Transmission of changes in light intensity. The on-off response. Many ganglion cells are specifically excited by changes in light intensity. This is demonstrated by the records of nerve impulses in figure. The upper panel shows rapid impulses for a fraction of a second when a light is first turned on, but decreasing rapidity in the next fraction of a second. The lower tracing is from a ganglion cell located lateral to the spot of light. This cell is markedly inhibited when the light is turned on because of lateral inhibition. Then, when the light is turned off, opposite effects occur. Thus, these records are called on-off and off-on responses. The opposite directions of these responses to light are caused, respectively, by the depolarizing and hyperpolarizing bipolar cells, and the transient nature of the responses is probably at least partly generated by the amacrine cells, many of which have similar transient responses themselves. Many ganglion cells are specifically excited by changes in light intensity. This is demonstrated by the records of nerve impulses in figure. Responses of a ganglion cell to light in 1. An area excited by a spot of light and 2. An area adjacent to the excited spot. The ganglion cell in this area is inhibited by the mechanism of lateral inhibition. The upper panel shows rapid impulses for a fraction of a second when a light is first turned on, but decreasing rapidity in the next fraction of a second. The lower tracing is from a ganglion cell located lateral to the spot of light. This cell is markedly inhibited when the light is turned on because of lateral inhibition. Then, when the light is turned off, opposite effects occur. Thus, these records are called on-off and off-on responses. The opposite directions of these responses to light are caused, respectively, by the depolarizing and hyperpolarizing bipolar cells, and the transient nature of the responses is probably at least partly generated by the amacrine cells, many of which have similar transient responses themselves. Retina and the central retina. For instance, a minute gnat flying across the field of vision is instantaneously detected. Conversely, the same gnat sitting quietly remains below the threshold of visual detection. Transmission of signals depicting contrasts in the visual scene. The role of lateral inhibition. Many ganglion cells respond mainly to contrast borders in the scene. Because this seems to be the major means by which the pattern of a scene is transmitted to the brain, let us explain how this process occurs. When flat light is applied to the entire retina, that is, when all the photoreceptors are stimulated equally by the incident light, the contrast type of ganglion cell is neither stimulated nor inhibited. The reason for this is that signals transmitted directly from the photoreceptors through depolarizing bipolar cells are excitatory, while the signals transmitted laterally through hyperpolarizing bipolar cells as well as through horizontal cells are mainly inhibitory. Thus, the direct excitatory signal through one pathway is likely to be neutralized by inhibitory signals through lateral pathways. One circuit for this is demonstrated in figure, which shows at the top 3 photoreceptors. The central receptor excites a depolarizing bipolar cell. The 2 acceptors on each side are connected to the same bipolar cell through inhibitory horizontal cells that neutralize the direct excitatory signal if all 3 receptors are stimulated simultaneously by light. Figure. Typical arrangement of rods, horizontal cells, H, a bipolar cell, B, and a ganglion cell, G, in the retina, showing excitation at the synapses between the rods and the bipolar cell and horizontal cells, but inhibition from the horizontal cells to the bipolar cell. Now, let us examine what happens when a contrast border occurs in the visual scene. Referring again to figure, assume that the central photoreceptor is stimulated by a bright spot of light while one of the 2 lateral receptors is in the dark. The bright spot of light excites the direct pathway through the bipolar cell. The fact that one of the lateral photoreceptors is in the dark causes one of the horizontal cells to remain unstimulated. Therefore, this cell does not inhibit the bipolar cell, and this allows extra excitation of the bipolar cell. Thus, where visual contrasts occur, the signals through the direct and lateral pathways accentuate one another. In summary, the mechanism of lateral inhibition functions in the eye in the same way that it functions in most other sensory systems to provide contrast detection and enhancement. The Hubel and Wiesel experiments greatly expanded the scientific knowledge of sensory processing. In one experiment, done in 1959, They inserted a microelectrode into the primary visual cortex of an anesthetized cat. Each of the neurons in the various layers of the retina covers an area in your field of vision. This area in space where the presence of an appropriate stimulus will modify the activity of this neuron is called the receptive field of this neuron. The receptive field of a single photoreceptor cell, for example, can be said to be limited to the tiny spot of light within your field of vision that corresponds to this photoreceptor's precise location on your retina. But in each succeeding layer of the retina, the receptive fields become increasingly complex, and they become even more complex when it comes to the neurons of the visual cortex. Here is an example of this complexity. The receptive fields of bipolar cells are circular. But the center and the surrounding area of each circle work in opposite ways. A ray of light that strikes the center of the field has the opposite effect from one that strikes the area surrounding it, known as the surround. In fact, there are two types of bipolar cells, distinguished by the way they respond to light on the centers of their receptive fields. They are called on-center cells and off-center cells. Just like bipolar cells, ganglion cells have concentric receptive fields with a center-surround antagonism. But contrary to the two types of bipolar cells, on-center ganglion cells and off-center ganglion cells do not respond by depolarizing or hyperpolarizing, but rather by increasing or decreasing the frequency with which they discharge action potentials. The organization of ganglion cells' receptive fields, composed of inputs from many rods and cones, provides a way of detecting contrast, and is used for detecting objects' edges. Each receptive field is arranged into a central disk, the center, and a concentric ring, the surround, each region responding oppositely to light. For example, light in the center might increase the firing of a particular ganglion cell, whereas light in the surround would decrease the firing of that cell. Stimulation of the center of an on-center cell's receptive field produces an increase in the firing of the ganglion cell. Stimulation of the surround produces a decrease in the firing of the cell. See figures. Stimulation of the center of an off-center cell's receptive field produces a decrease in the firing of the ganglion cell. Stimulation of the surround produces an increase in the firing of the cell. See figures. Stimulation of both the center and surround produces only a mild response, due to mutual inhibition of center and surround. See figure. Transmission of color signals by the ganglion cells. A single ganglion cell may be stimulated by several cones or by only a few. When all three types of cones, the red, blue, and green types, stimulate the same ganglion cell, the signal transmitted through the ganglion cell is the same for any color of the spectrum. Therefore, the signal from the ganglion cell plays no role in the detection of different colors. Instead, it is a white signal. Conversely, some of the ganglion cells are excited by only one color type of cone but inhibited by a second type. For instance, this frequently occurs for the red and green cones, with red causing excitation and green causing inhibition, or vice versa. The same type of reciprocal effect occurs between blue cones on the one hand and a combination of red and green cones, both of which are excited by yellow, on the other hand, giving a reciprocal excitation-inhibition relation between the blue and yellow colors. The mechanism of this opposing effect of colors is the following. One color type of cone excites the ganglion cell by the direct excitatory route through a depolarizing bipolar cell, whereas the other color type inhibits the ganglion cell by the indirect inhibitory route through a hyperpolarizing bipolar cell. The importance of these color contrast mechanisms is that they represent a means by which the retina itself begins to differentiate colors. Thus, each color contrast type of ganglion cell is excited by one color but inhibited by the opponent color. Therefore, color analysis begins in the retina and is not entirely a function of the brain. Thank you for attention.